I'm Gary Gilliland. I'm a professor of medicine at Harvard Medical School with appointments at Brigham and Women's Hospital and the Dana-Farber Cancer Institute. In the context of the Harvard Stem Cell Institute, I'm the director of the Cancer Stem Cell program that has as its goal the development of better treatments for cancer based on an understanding of the stem cell nature of cancer.
That's basically the status.
Well, that's good.
Right
It
now.
looks promising, but
I think
this
so.
is fantastic to get this kind of done.
Yeah. No, I'm very happy with that.
And
Absolutely.
how many did you? Where did you get those hairpins from?
Open Biosystems.
Really?
Yeah.
You just tried a bunch of them? We treat many cancers today the same way that we treated them 20 years ago, with cytotoxic chemotherapy. It has lots of side effects, and with very few exceptions, we can reduce the size of tumors, but we can't eradicate those tumors. We can shrink ovarian cancers, we can shrink many types of lung cancers, we can shrink colon tumors, we can shrink tumors affecting the central nervous system, as has been done for Senator Kennedy. But if the tumor is metastasized, if it's spread out of a site where you could surgically remove it, for the vast majority of solid tumors, we simply can't cure them. We can only reduce them. We believe that the reason that we haven't been more successful is that we may not be targeting the right population of cells, the so-called cancer stem cells that ultimately are responsible for the propagation and the relapse of tumors after initial response to therapy. I think the most important concept to take home is that every tumor that we treat has potential for self-renewal. It has properties of stemness, and whether that's one cell in a million, or it's one cell in 10, or one cell in 2, that's the property that we need to be attacking to cure cancer. There's no question that that property exists in all tumors for which we don't have cures. They come back, they're being spawned by a cell that has properties of stemness, that it is immortal, it can renew itself, and it can propagate the entire tumor in its entirety, just the way a root can propagate an entire weed if you give it time and nutrients. Does it correlate with efficiency of excision, or is there a relationship between those
It does.
two?
There is, yeah. And so basically something that we had talked about before was looking at is, when we knock out the gene, is it causing increased cycling of the cells?
One of the projects that we have that's supported by philanthropic efforts through the HSCI is what we call the Cancer Stem Cell Project. So this effort allows us to take advantage of this extraordinary capability we have here, which is that we've got some of the best surgical oncologists in the world. I don't want to be controversial about it. There's many other good surgical oncologists around the country, but we've got some really good ones here, the Mass General at the Brigham, and we can tap that resource to get primary tissue, which is going to be the driver. But it's very expensive. It's not the type of thing you can get support from the federal government to do for the most part. So that's been represents a terrific opportunity that we're trying to take best advantage of. We're focusing a lot of attention on leukemia stem cells because they're so well characterized. And we have a fantastic collaboration that rose out of the HSCI with David Scadden, who's at the Massachusetts General Hospital. Young investigators in his lab and mine have developed this fantastic screening platform. If you have a group of compounds, let's say 5 or 10,000, that you'd like to screen and see if any of these kills cancer stem cells, you need to have throughput mechanisms that involve robots to dispense cells and educated microscopes that can interpret the results from the assay system. But most importantly, you have to be able to propagate the stem cells in a dish and have them retain their properties of stemness. So the challenge is, how do you translate that observation into a high throughput automated robotic platform that allows you to screen for tens of thousands of drugs? And that's the platform that we've developed. It's very exciting. We're just now to the point where we're initiating screens. And his laboratory, which is expert in normal hematopoietic development, is screening for compounds that have effects on normal stem cells. And that could give us ways of expanding normal stem cells, which has been a holy grail. We'll identify compounds that will kill normal stem cells that we'd probably want to stay away from. And we're doing a parallel screen in our lab with expertise in leukemia stem cells, looking for exactly the same characteristics. What things cause these to grow? Of course, we want to stay away from those. And what things kill the leukemia stem cells? And then we cross-collate the data and pick those that kill the leukemic stem cells and don't have an effect on the normal stem cells. It's a very interesting program within the context of the broader goals of the HSCI, because most of the goals are focused on regeneration, taking stem cells, expanding them, using them for good purposes. Can Doug Melton make a pancreas or an islet cell that would help treat diabetes? Can we make kidneys? Can we generate a heart using stem cells? Those are all very exciting areas in regenerative medicine. They all focus on growing stem cells, helping them expand into progeny that could be heart muscle or it could be skeletal muscle. We have the opposite problem. We want to understand stem cells so we can figure out how to kill them. I give Doug Melton and David Scadden a lot of credit for the genesis of the concept, the idea that we should all be working together, being mutually supportive, and tapping the expertise that we have in a way that's mutually supportive and synergistic to try to understand stem cell biology from a very broadly based perspective.